Thank you for joining us, everyone. We're going to give it a couple of minutes to fill the room here. Thanks again, everyone, for joining us. We're going to give it about another 30 seconds or so to fill the room. Thanks once again for joining us. We'll give about another 15 seconds here to fill the room up. Well, good afternoon, everyone. Thanks for joining us once again for our weekly Wednesday webinar series. Today's webinar is on using math to understand bleeding in hemophilia patients. My name is Brett Spitali, and I'm the Vice President of Advancement here at the National Hemophilia Foundation. At any point during the webinar, if you would like to ask a question, please use the Q&A feature at the bottom of your screen. We have NHF staff members monitoring these questions, which we will pose to our presenter after the presentation. This webinar is being recorded and will be available to the community beginning on Friday, June 4th. I'm joined by Dr. Corinne Lederman, who is the Associate Professor and Director of Graduate Studies in the Department of Applied Math and Statistics at Colorado School of Mines. Thank you for taking the time to join us today, Dr. Lederman, and I'll now turn it over to you to get us started.
Great. Thank you so much for the invitation. These webinars are always interesting. I know there's a lot of you out there, although I can't see any of you. But I hope that my talk today is of some interest to you. And if you have any questions at all, please feel free to ask. So as was just said, I'm going to talk today about how I use mathematics to try to understand bleeding, and specifically variation in bleeding phenotype in hemophilia. And we'll get into that more in a little bit. So just a brief outline. Today, I'm going to start with a refresher of the blood clotting system and highlight its complexity, which is really my motivation for using mathematics to study it. And then I'll talk about a specific question relating to bleeding in hemophilia that we're trying to answer using a mathematical approach. And I'll talk about some of our mathematical results and some experimental results that we use to validate some of the mathematical ideas that we came up with. And then I'll summarize where we are and where we're going next with the research. So blood clotting is a basic defense mechanism for all vertebrates, right? All animals with backbones, including mammals, birds, reptiles, amphibians, and fish. And most invertebrates actually also have a clotting mechanism. But we as humans want to keep our blood within our closed circulatory system, as we need it to transport oxygen and other nutrients around the body. In short, we need a quick, efficient way to stop the leakage of blood that is localized to the site of injury where there's bleeding. So among vertebrates, there's a few differences in the clotting system, and it depends really on environment. For example, hibernating squirrels, or having different organ systems, etc. Clotting system didn't start out that complex, but it has evolved over time to become more and more complex, likely to regulate animals' responses under a wider spectrum of environments. For example, species have evolved to have higher blood pressure, more complicated cardiovascular systems, higher metabolic rates, and new organs and organ systems. And the clotting system had to evolve along with all of these changes. So the clotting system is thought to have begun as a small system of a few chemical reactions and physiologic interactions, sort of illustrated in this little schematic I made here at the top right of the slide, where there's just a few interactions happening. But over time, it has since evolved into something more like what you see down here on the bottom right of the slide. And I'm not going to expect people to know every single step in this diagram, and I'll talk through some of them in more detail as I work through the talk. But the point here is just to show, again, the complexity of the clotting system and the coagulation system. So there are some common characteristics of the clotting system within all vertebrates. First, there are cells that aggregate to loosely plug the hole as an initial phase of clotting to stop or slow bleeding. Then there are interactions between many proteins, usually in the form of enzymatic reactions, to amplify and further propagate those chemical reactions. Cellular surfaces are necessary to further many of these reactions, but they also help to localize the proteins to the site of injury by allowing the proteins to bind their surfaces and be used exactly where they're needed, right, at the site of injury. Then a fibrous mesh forms as a result of the chemical reactions, and the mesh stabilizes those clumps of cells. And all of this is occurring under flow. So let's translate this into the human clotting system, where the normal healthy process is called hemostasis. So there's two stages, primary and secondary. Primary hemostasis is where the cells do the clumping. And in humans, those cell clumps are platelets. Okay, and then next stage, in secondary hemostasis, this is where the protein interactions take place. And these are known as the coagulation reactions. And they occur on platelet surfaces. And through those reactions, an enzyme thrombin is created, and it helps to form a fibrin mesh that then holds that platelet aggregate together. And in hemophilia, the deficient clotting proteins affect the coagulation reactions in a way that results in a decreased amount of that thrombin to be generated, and thus there are leaky and unstable clots that form. All right, so to set the stage for the rest of the talk, I'm going to walk through this overall process just a little more slowly. So first, this is just a schematic of a blood vessel lined with an intact layer of endothelial cells. That's what you see here shown in yellow. And these are the cells that act as a barrier to keep your blood from clotting all the time. The purple ovals here are meant to represent platelets, and the blue circles are what are known as zymogens, which you can just think of as precursors to some of these enzymes that are going to be used later in the coagulation process or the clotting process. They get converted into enzymes later. So when there's some sort of injury or mechanical disruption to the vessel wall, cells in the subendothelium, that's just the layer underneath the endothelium, become exposed to the plasma. And within that tissue, there's tissue factor and collagen that is now making contact with whatever's in the blood. And notice on this picture, right, I don't have any blood actually leaking out of this vessel. This is just for schematic purposes to kind of show all of the reactions and sort of interactions here. But of course, in an injury where there's bleeding, there would be an actual hole and blood would be leaking from the vessel. So certain clotting factors in the plasma will then bind to the tissue factor and initiate this, what we know as blood coagulation, which is a large biochemical network of reactions, and I'll talk more about that in a minute. And in addition, the platelets then can adhere to the collagen and become activated. When the platelets get activated, they secrete chemicals from within them that can activate other nearby platelets. And they can also begin to support chemical reactions on their surfaces. So here again, here comes those cell surfaces again that are really important in the process. They also become sticky and they want to aggregate together and form a clump. Okay, so let's just briefly zoom in and look at the platelet aggregation and coagulation in a little bit more detail. Okay, so on the left bottom here, this is just a little picture of activated platelets that adhere to the subendothelium and aggregate together through a bunch of complex interactions with different receptors on the surfaces and other proteins. And coagulation, here's this picture I showed a few slides ago, sort of zoomed in, is this complex network of chemical reactions that involves all of these clotting proteins or clotting factors. And as many of you know, right, the notation here is to use Roman numerals for the clotting factors. So obviously factor 8 and factor 9 are the deficient proteins in hemophilia A and hemophilia B, respectively. So as the process continues, the platelets aggregate and coagulation continues at the subendothelium. Enzymes are created at the subendothelium. They can move through the plasma and begin to react on the platelet surfaces to produce the final enzyme, the final coagulation enzyme known as thrombin. And I'm going to talk a lot about thrombin today, which is why I wanted to walk through the steps. Thrombin then cleaves another molecule in the plasma, fibrinogen, into fibrin, which then polymerizes and forms a gel that holds all those platelets together. And all of this together forms a stable blood clot. And finally, just to point out that all of these processes are occurring under flow. So now let's just imagine a small injury that's occurring under flow. So let's consider that platelet aggregation and blood coagulation are occurring in a very small reaction zone here, as I've drawn in this black box, above some injury, and that the proteins in the platelets are being supplied to and removed from this reaction zone by flow. So this system under flow is not really as intuitive as you might think, right? Flow provides the supply and the removal, but this can enhance some reactions by providing almost constant levels of all the things that are needed for the reactions to occur. But it can also attenuate or lessen those reactions because it's also diluting that reaction zone, right? By taking things away. So this kind of brings us to the motivation for mathematical modeling, right? The complexity and lack of initial intuition is precisely where mathematical modeling can be useful. So maybe a mathematical representation of this process can help us sort of wrap our heads around this complexity in a systematic way. So the goal here is, the goal for me and the goal of the study I'm going to tell you about today was for us to try to answer specific questions about the clotting system by using mathematical models that offer mechanistic insight and testable hypotheses. So I will just skip that. All right, so the specific question I'm going to talk about today focuses on hemophilia A, the bleeding disorder characterized by the deficiency in clotting factor 8. Probably the facts on the slide are mostly known by this audience, but I'll just go over them again. So hemophilia A is a genetic condition that's inherited in an X-linked recessive pattern. And deficiencies are categorized by percentage of measured factor levels as mild, moderate, and severe. So the main motivation for the work that I'm going to talk about today was to try to understand the well-known variability of bleeding phenotype in hemophilia A that results in factor levels alone being a poor predictor of bleeding risk. So interestingly, about 10% of patients with severe hemophilia rarely bleed, and the mechanisms underlying this phenotypic heterogeneity is currently unknown. And so this led us to ask the following question. In cases of hemophilia A with severe deficiency and mild bleeding, what is compensating for the deficient clotting protein? What else is going on in the system that's allowing there to be only mild bleeding rather than severe? And so we wanted to ask the question, well, what about other clotting factors? What's going on with the other clotting factors, and how might they be also contributing or playing a role to this variability? So let's talk a little bit about normal variation of other clotting factors in the body. So it's well known that the plasma levels of clotting factors vary among individuals. The variation that's considered normal and still healthy is a range between 50 and 150% of the average value of the healthy population. So I've created a little schematic here on the right. This is not data. This is just something that I've drawn to illustrate this idea in kind of a simple way. So the center line within each of these purple boxes represents the average or the mean of the plasma level concentration of each clotting factor. So down here is a list of the clotting factors, factor 2, 5, 7, 8, 9, 10, and 11 here. And then the height of the box represents the spread between 50 and 150% of that mean. Okay. Thus, so we hypothesize that there could be clues to our question regarding the mechanism of phenotypic variability. Could there be synergistic or antagonistic behavior between other proteins in the system? In other words, could 50% of one protein, of one factor, combined with, say, 150% of another factor, but both still within the normal range, work together to enhance the clotting response, right? Could it be more than two? Would they be in a low normal or high normal range? So the ultimate question we wanted to answer then is, how do variations in plasma levels of other factors alter thrombin generation when factor A is deficient? And not necessarily variation of one factor, but variations of multiple factors simultaneously. And maybe a related and possibly more important question would then be, can we get or create sufficient amounts of data that we can then statistically analyze to answer this question? So what is necessary for generating such a data set? So such a data set is not so feasible to create. So let's see a quick calculation to see why. So if we just look at these first 5 clotting factors here in the red circle, and we think about wanting to vary each of those between a low normal, normal, and high normal level, we're going to have immediately, we come up with 243 different combinations of these factors and their levels. That is a lot of different combinations to have to go through, and that's only for 5, right? And we might have more and maybe other proteins that we also want to investigate. And now imagine varying all these factors in an experimental setting with replicates, right? Because you want to do your experiments a number of times. These experiments could be of different types. They could be monitoring fibrin deposition and platelet deposition in what's known as a microfluidic assay, where blood is flown over tissue factor and collagen patches, and the clotting is monitored through microscopy. Or it could be in the form of looking at thrombin generation assays, where you have a test tube with cells and plasma, and you are looking at how much thrombin is generated within that test tube. So more like a clinical assay. So a small number of experiments are certainly doable, but the number of experiments that would be necessary to create such a data set is just not feasible, right? So we came up with another idea of how we could generate a large data set that we could study. How about using a mathematical model to create a large synthetic data set? So over the last 2 decades, our group has developed and refined a validated, dynamic, mechanistic, mathematical model of flow-mediated coagulation. There's a lot of words there. Let me go through them. So the model is mechanistic because it considers all the interactions between clotting factors, flowing blood, platelets, tissue factor, etc. It's dynamic because it tracks changes in all of these things in time. So it takes inputs such as plasma levels of factors, the flow rate, etc. All of these affect relationships within the inner workings of the model, just drawn as this little sort of gray box here, and tracked with mathematical equations that describe how the concentrations of everything changes in time. And then the model outputs concentrations of any species in the model as a function of time. Okay, and so an important output that we want to look at is thrombin, right? Because I said we really want to understand how thrombin generation is being altered or changed. So this model can be simulated in seconds of real time on any old computer, and thus it's incredibly efficient and easy to test out hundreds of thousands of potential variations in all these clotting factors. So a systematic variation of the plasma levels of clotting factors is feasible and can serve as an initial screening over potential modifiers of thrombin generation. So just a quick note about this little drawing I made here. Our model simulates coagulation under flow, right? This is why I wanted to talk about flow quite a bit throughout the talk so far. It's kind of been a theme. But the thrombin generation in this model is often sustained in time. So this is, you can see this here by these curves kind of going up, and they sort of stay up, where this is the thrombin concentration as a function of time. So this is due to that fact that there's a constant supply of substrates or proteins that are being brought into the system. So I like to point this out here because this is different than what you may have seen in a thrombin generation assay, where the thrombin concentration goes up and then comes back down, because there's a limited amount of proteins in a test tube, right? It's a closed system, and all of the thrombin that gets produced in that system gets then inhibited or inactivated by antithrombin. So we'll be looking at some model output, and the thrombin curves look more like this, where the thrombin concentration is sort of sustained in time. And this is why I wanted to point out these characteristics here before we start looking at the model data or the model output. So I'll just give a big picture of the model here. I'm happy to discuss details if you want to discuss details after the talk. The model we've used for this study simulates that reaction zone that we talked about a few slides ago, right? So the reaction's occurring in this small zone above some injury where there's exposed tissue factor and collagen, with everything subjected to flow. So we can write down a differential equation that tracks the concentration of every species that exists in the reaction zone. All of those zymogens, enzymes, platelets, and things that are bound to the platelets and bound to the subendothelium too. You don't need to know what a differential equation is necessarily. All you really need to know is, well, you don't even need to know this, but I'll just explain the form, right? All these equations have a similar form, where the rate of change of a concentration in time is just set to be equivalent to the rate that something is coming into the zone minus the rate that that thing is moving out of the zone, and then plus or minus whatever is being generated or inactivated within the zone. Okay, so it's just this balance of rates. Okay, and then we can write down this differential equation that looks kind of like what you see here on the slide. So there's numerous model inputs that describe this scenario that we want to simulate, right? The inputs here, as I said, are the clotting factors, kinetic rate constants that define how the reactions are occurring, the platelet count, the flow rate, etc. And all of these go into the model, and then the equations of the model are solved numerically, and the solutions to these equations are the concentrations of every species in the model. And then the solutions can be visualized by plotting them against time, okay? For example, thrombin. So again, I'll point out here that the thrombin is seen here as a sustained response in time, okay? And even though we're only looking at thrombin here, there's actually about 86 different species that we track in this model, and we can look at any of them or any combination of them, which is one reason that the model is just so powerful. So now we have a question we want to answer, and we have a model that we like. And so our first step in this sort of research pathway or process is to use the model and perform hundreds of thousands of simulations with all these various inputs of plasma levels of factors to generate the large synthetic data set. And I'll move through this diagram and sort of add on as we go forward. Okay, so I'm going to stay on this slide for quite a while, so let me see if I can talk through it. So let's take a look at what kinds of output the model generates. So what you see here on the right in this plot is called a quantile plot of the thrombin curves as a function of time generated from 100,000 simulations of our model, where we fixed factor 8 to be low, and then we varied all the other clotting factors between 50 and 150% of normal. What you see here is time, again, on the x-axis down here, and thrombin concentration on the y-axis here. And this is actually on a log scale, but that's okay. You don't need to pay attention too much to that. And this is going out to 40 minutes of time. So let me explain what this quantile plot is telling us. So what this plot tells us is that about 90% of all of those 100,000 simulations, all of those lie between here, this orange and pink region. So most of the simulations gave us all the curves that would lie within this range, okay? The maximum value that came out of all these variations is shown by this gray dashed line, and the minimum of all these simulations is shown in this gray dashed line down on the bottom. And then between the orange line and the gray dashed line in here is about 5% of all the simulations. Okay, and down here is another 5% of the simulations. And remember, there's 100,000 of them. There's a lot of different variations that we did. And this blue line here, this is equivalent to 1 nanomolar. It's just a specific concentration of thrombin. And this is a really important concentration in our model. It's an important measurement because it's at this level that platelets start to get activated. And it's at this level where we know if the model gets to this concentration, the system will go, so to speak. It will fire, it will burst. There will be a large thrombin response after that. So maybe you recall in some of the slides previously when we talked about these, characteristics of clotting systems, these protein interactions are, right, there's enzymatic reactions for amplification of the system. So this amplification is really important in clotting, right? Because you want to be able to clot enough when you need to, but not so much when you don't need to. So there has to be some sort of threshold the system can get over, right? So that you're not clotting all the time from some small injury in a normal healthy person. Okay, so interestingly, only about 4% of the simulations, right up here, led to thrombin levels above 1 nanomolar after 40 minutes of clotting. And I'll note here that these were simulated with a low level of tissue factor. You can just think about that as being just a small sort of injury size or a small initiator of the injury. And we did this because we actually did not expect to have a strong thrombin response in most of these variations, because this is a simulation of hemophilia. And so with a low tissue factor level, we would not expect normally to see a strong thrombin response. What we're asking here is, are there combinations of all these factors that push the system above that threshold and enable it to make more thrombin than we would expect? And that's exactly what we see here, is that we see that about 5% of these top simulations made more thrombin than all these other ones. And so our question is, what about these? Which combinations of those model inputs led to those top 4% of producers or simulations? Okay, so we're going to zoom in and we're going to look at those 4,000 simulations, and we're going to try to extract information out of those to help us answer that question. So the next stage of the process is to take the output and statistically interrogate it to potentially yield modifiers of thrombin generation in hemophilia. So let's go back to this picture. We're going to focus in on these 4,000 simulations, and we want to provide a descriptive statistic for what we are seeing in this output. Okay, so what we did is we extracted the distributions of the model input in the form of the clotting factor, the plasma levels of the clotting factors that led to these concentrations. So these extractions are quantified in the box and whisker plot you see here on the right. And you don't really need to know too much other than you can see that 2 are not like the other, right? 2 of these are showing very high skew in these distributions. If everything was sort of on average doing the same thing, we would see all these blue boxes sort of lined up across the middle, and we don't see that, right? We see that there are 2 of them that are doing something a little bit different. And in particular, we see that the skew shows that high normal factor 2 or prothrombin and low normal factor 5 here enhances the thrombin generation in hemophilia A. So this was our statistical extraction from this huge data set. And now we want to know, well, can we further test just these two factors in the model? So let's fix everything else and let's just look at varying factor 2 and prothrombin and see if we actually see what we think we see. So to test these two factors, one low normal and one high normal, we will have 4 specific cases to investigate, as shown in this little table here. So let's walk through the results of each case. So first, we see that the normal levels of factor 5 and prothrombin, that's here, and this is shown as the blue curve. We have a very weak thrombin response, and in fact, the concentration begins to decrease at about 25 minutes, and that's just shown by this curve sort of starting to come back down after some time, right? It doesn't have that bursting behavior like I mentioned before. So now if we keep the levels of factor 5 normal and just increase the level of prothrombin, the results are shown here in the black curve. We still have a weak thrombin response that does not meet that 1 nanomolar thrombin by 40 minutes. If we then decrease factor 5 to a low normal level and keep prothrombin normal, we see that there definitely is an amplification and a burst is produced. You can see that because the curve is starting to increase there at the end. And it's just by 1 nanomolar by 40 minutes. Whoops. Then the combination of the low normal 5 and high normal prothrombin produces a robust thrombin response. And this kind of confirms that result of our descriptive statistic, showing that just these four variations, we should be able to determine, right, if low normal 5 and high normal prothrombin enhance thrombin generation in hemophilia A. So this brings us to the next stage of the process. I mentioned we want to have a model, we want to make a hypothesis, and we want that to be testable. And that's exactly what we have generated here. We want to propose, let's look at these 4 different cases in an experiment and see if we can validate what we saw in the model. So to test the hypothesis, we used whole blood microfluidic assays, okay? So here, whole blood from individuals with moderate and mild factor VIII deficiencies were flown over surfaces embedded with tissue factor and collagen at a shear rate of 100 inverse seconds, just relatively slow flow. The blood was treated with exogenous prothrombin, so we added prothrombin to modulate that concentration, or an anti-factor V function blocking antibody that reduced the factor V activity to about 60% in normal pooled plasma. So it's really hard to just remove a certain amount of factor V, right? So we use an antibody that essentially blocks the function of factor V. So it doesn't allow some fraction of the factor V in the blood to do what it normally would do. And then the 4 conditions that I suggested from before were tested in this assay. And I'll just point out here that the thrombin in our model is used as a surrogate for fibrin deposition in the assay. So you're going to see green as platelets and red as fibrin in the pictures I'm going to show you in a minute. And we're going to be comparing the qualitative behavior of the fibrin to our thrombin outputs from our model. Okay. So let's go through some pictures. So first, with no modification of factor V or prothrombin, we see a little bit of platelet aggregation, but little to no fibrin. But this is expected from hemophilia A blood, right? We know that we're affecting the coagulation reactions, we're affecting the secondary hemostasis, so we should expect to see platelet aggregation, but not a lot of fibrin deposition or no fibrin deposition at all, which is what we see. Then with the addition of some prothrombin, there was slightly more platelet aggregation, which suggests a slightly higher thrombin, but maybe enough to activate some platelets, but not enough to start making large amounts of fibrin. Then with the addition of the anti-factor V function blocking antibody, this resulted in more platelet aggregation, and we start to see fibrin deposition become visible and measurable. And finally, with the addition of the antibody and prothrombin, we see larger platelet aggregates and a very increased level of fibrin deposition. And so in summary, we were able to observe experimentally that partial inhibition of factor V and increased prothrombin enhanced the fibrin deposition of hemophilia A blood under flow, which validated our hypothesis that was generated from a mathematical model. So then we further tested this model hypothesis using thrombin generation assays by way of calibrated automated thrombography, or CAT. And here we see that results using factor VIII deficient plasma, less than 10% of factor VIII, treated with vehicle or added prothrombin or the same anti-factor V antibody, and prothrombin plus the antibody. So we can see that the partial inhibition of factor V increased the thrombin peak. So if you look at the blue curve here, this is the vehicle. And if you look at the orange curve, this is just the addition of the factor V antibody. And you can see that the maximum point of these curves has been increased. So that's the peak value of thrombin. And then there's even more when you add prothrombin to that, in addition to the anti-factor V antibody. And so this is just one more line of evidence that suggests that low normal levels of factor V enhance thrombin generation when factor VIII is deficient. All right, so why does this work? Why does a reduction in factor V enhance thrombin generation when factor VIII is deficient? Well, this brings us to another stage of our research pathway, which is we can use this model for focused discovery of mechanisms, right? We made a hypothesis and we tested that, but why does it work? Why does it happen? So this is available to us because, remember, our model gives us access to every concentration of every species in the model, and we can probe these outputs to determine what led to the thrombin enhancement. So due to the time of the talk and just sort of the sort of technical aspects of the model, I'm just going to walk through the word story that we were able to uncover with further exploration of our actual mathematical model. And the details of these dynamics, I can refer you to the paper here that's on the bottom of the slide, but hopefully you'll understand the mechanism as I walk through it just with words. Okay, so back to this question, why does this work? And then I'm going to pop up this picture of coagulation again. So under normal non-hemophilic conditions, factor V and factor VIII are typically considered to be procoagulant, right? We need them. You need them to clot properly, right? And they aid in the thrombin generation. Factor V and factor VIII in their activated forms serve as cofactors for other enzymes that are necessary for generating a large amount of thrombin, okay? So those are just kind of shown here in green. So we know factor V and factor VIII are necessary. Their activated forms are necessary. We need to have thrombin, but so what else can we think about with these two proteins? Well, there is a clue. And one clue is that factor V and factor VIII are both activated by the same enzyme early in the coagulation process, factor Xa. So this little schematic is trying to show that. So here's factor V and here's the enzyme, and it activates it to its activated form, factor Va, and it activates factor VIII to its activated form, factor VIIIa. And so this is interesting, right? Because we're saying that in hemophilia, we have a low level of factor VIII, right? And we need it to be activated in order to further this process and get thrombin generation. But factor V is another protein that we need, but it also needs to be activated into its active form. And these two things have to be activated by the same enzyme early in the process, okay, to get the system going. So the proposed mechanism is a competition between these two for that enzyme. So what I mean by this is if you reduce the factor V levels, you're weakening the competition between it and factor VIII for being activated by that enzyme, right? So the result is that more of that already small amount of factor VIII can become activated, and so more is available to serve as a cofactor to amplify the other reactions and lead to thrombin production. So we explored this idea using the model and can prove that, at least within the mathematical model, this is the mechanism for enhancing thrombin generation with low normal factor V, that it's this competition story between 5 and 8. There are other potential mechanisms that could be at play. And here's a few that I'm not going to spend too much time on this slide, but I'll just say there's a summary of 3 potential mechanisms we think could enhance thrombin generation. So the first row here, this is what I mentioned on the last slide, which is what our model in the current form suggests. The other 2 mechanisms are related to a protein called protein C, and in its activated form, activated protein C, and tissue factor pathway inhibitor, or TFPI. These are 2 other proteins that inhibit the clotting system normally. So the mechanisms by which they could potentially affect the system that I talked about today are not currently included, or were not included in the model in a way that could potentially affect the system in this way. So these are mechanisms that we thought about after the fact and need to further explore. So we are adding some of these things into the model to be able to explore these as other mechanisms. That being said, we have preliminary biochemical experiments, not published yet, from our collaborator that does suggest that this substrate competition mechanism is at least in part a contributor to this thrombin generation enhancement. But there's more work that needs to be done, and we're very excited to keep working on this, and also in looking at other hemophilias as well. So in summary, mathematical models are useful for answering many questions about the complex blood clotting system under flow. Our model here helped to facilitate discovery of a previously unrecognized interaction within the clotting system. We made a prediction with the model. We tested and validated this prediction with experiments. We then used the model to determine mechanism, and we're currently trying to test that mechanism experimentally. And we're also working on using the same approach to investigate hemophilia B and hemophilia C. And with that, I'll just acknowledge all the amazing people that I've gotten to work with over the years, all the students that work so hard on all this work, all my collaborators at different institutions, and of course, my funding from the Army Research Office, the National Science Foundation, and the National Institutes of Health. And with that, I will stop and take any questions that you might have. Thanks for listening.
Thanks so much, Corinne. We appreciate it. We do have several questions that came in, and I'll just start right off the top here. First question is, could this model or math be used to describe other inheritable blood disorders such as VWD?
Yes, absolutely. We currently do not represent von Willebrand factor in a super detailed way in the model. Von Willebrand factor is a very complex little molecule, but it definitely can be studied, and we are working in various groups to start adding in the biophysical, correct biophysical representations of how von Willebrand factor works. One idea that we have to start to get going on that is to sort of include it in a simple way that affects the way the platelets aggregate with one another, because that's really where the von Willebrand factor comes into play. It helps the platelets aggregate, it helps the platelets stick to different surfaces and things like that. So we're trying to think about some simple ways to get started by just modulating some of those rates without explicitly modeling all the interactions of von Willebrand factor. But we definitely are interested in that and hope to work on that some in the future.
Great. Thanks, Dr. Lehrman. The next question that comes in, obviously the real world is complex with many interrelated components, whereas a mathematical model is an abstraction of reality. How are you addressing the influence of the real world in this model?
Yeah, I mean, that's a great question. And I think our model is already actually including more of the real world than a lot of other models that are out there. Most mathematical models of clotting have focused only on these test tube models without thinking about the biophysical and biomechanical interactions with platelets and flow. They're very important. Most other models don't even consider platelet surfaces at all. They just consider the reactions happening in the plasma. But we know over the last decade that cell surfaces are extremely important in regulating the clotting system, and things just happen differently under flow. So I would definitely agree that this is an abstraction, but I think we're actually making a lot of progress in terms of incorporating more realism and more sort of realistic features in this model than previous models that have come before us. And also, what we've been trying to do with this model, and there's other versions of it that are even more complicated that can model larger injuries and start looking at things happening in space as well, is we work very closely with engineers and clinicians too. And one interesting thing about this model is that it actually works really well with these microfluidic assays. And so what I mean by that is we can have the model and the assays talk to one another. And even though that's not an animal or a human yet, it's a great place to start because it's something where we can look at the model in an easy, cheap way and then go ask, can we look at this in the in vitro system? Can we look at this in experiments? And maybe we can validate this hypothesis. And then from there, we can go on and ask further questions about things happening in vivo. And I think that's what was so exciting about this particular project, is that we were really able to have the models, experimental and mathematical, kind of work together to look at things that are more realistic. That was kind of long-winded, but I hope that answered the question.
Absolutely. Thanks, Dr. Lideman. The next question that comes in, could levels of thrombin be a better indicator of bleeding risk than factor VIII levels in patients with hemophilia?
Well, I don't think I'm the right person to say yes or no on that. I think I probably not yet. I mean, I think thrombin generation is certainly something that people look at, but I don't think that there's consensus yet on how to look at how to correlate bleeding risk necessarily with thrombin generation assay results. I think it's still a challenging thing to understand how we can say what bleeding is by looking at a test tube model of clotting and thrombin generation. What I hope will happen is that through the years we'll try to better understand what bleeding really is and what it means in different parts of the body. For example, right, in hemophilia, there tends to be more bleeds in certain areas of the body than others. Say when people are taking anticoagulants and bleed. And why is that? So what's different about the different tissues? And I still think there's a lot to learn about what bleeding is, but I don't think at this point, thrombin generation, at least not in the clinical assays that we have, I don't think there's consensus on that correlating directly with bleeding risk. But maybe someone else in the audience can answer that better than I can.
Thanks, Corinne. The next question that comes in, so why do patients with combined factor V and factor VIII deficiency have severe bleeding manifestations? What about patients with factor V short?
Excellent question. So the combined factor V and factor VIII bleeding disorders are both really low levels of factor V and factor VIII. So remember that what I'm showing here is factor V levels that are still within the normal range. They're still above 50%. So as part of this, in the study that we published, we did reduce the levels of factor V down to very, very low levels. And at some point, the factor V gets so low that you don't that you can't clot, you won't make thrombin. So this is a really interesting result, actually, because it's sort of this nonlinear effect, but certainly below some levels of factor 5 and factor 8, you won't clot. But what we're saying here is that if you decrease it from normal, it can actually enhance this. It can actually enhance this, this response. And in terms of factor 5 short, so that third mechanism I was talking about on the last slide, factor 5 short is a different isoform of factor 5 that can be inhibited or bound to by tissue factor pathway inhibitor. So this is something we're currently actively working on and actually pretty excited about, because our preliminary results are showing that this inhibition via factor 5 short with TFPI, we still see the same competition, substrate competition story at play, but it's actually enhanced more with variations of TFPI, which is not what we saw in the data that I showed today. But more to come on that. But it's definitely, TFPI is going to be playing a role via the factor 5 short.
Great. Thanks, Corinne. Appreciate that. The next question that comes in, how does the mathematical model take into account the variations in mutations? Is there any anticipated difference?
Right. I mean, I guess what I'll say here is that, we're simulating things just on short timescales, tens of minutes, 20 minutes, 40 minutes. We're not necessarily looking at any mutations that would occur on such timescales, of course, but we can definitely alter characteristics of the proteins in any way. Now, we haven't done that here, but certainly if there's a mutation that changes the binding characteristics or the enzymatic, the catalytic efficiency or anything like that, we can definitely incorporate those things into the model and see how the model behaves. We haven't done that for any specific cases yet, though.
Great. Thanks again. The next question that comes in is, have you identified any limitation of your mathematical model? If so, can you explain these?
Yes. Well, I think it does come back to this idea of it's a very complex system and we can't include everything. And they're definitely, for this particular model that's very, very efficient and fast, it is so because we assume that the injury is very, very small. And so because the injury is small here, we can make some mathematical assumptions about what types of equations we can use to represent the system and what we have to solve. If the injury gets much bigger, the mathematics gets much more complex because you have to start tracking how things move around in space. And that just takes more computational power, more mathematical power. We do have something like that in place, but it's slow, right? It takes days to run, perhaps, rather than seconds to run on a computer. So that's definitely one limitation of this version of the model. But again, we like to use this as kind of a screen for interesting artifacts or interesting outputs. And then we can use those and kind of explore them in a more computationally expensive, if you will, type of simulation. And then also, I mean, there's a lot of different proteins that are out there that people are discovering new roles for. And there are people that work on one clotting protein for their entire career. And there may be so many little intricacies about those proteins and how they work that, our model may not incorporate every single thing. And I think that can be challenging, but at the same time, we can incorporate lots of different things. So as the research is growing and building, like it has been with the literature on factor 5 short, for example, over the last decade or so, we can start to incorporate these things as the research comes in. And I think that's pretty exciting.
Great. Thanks, Dr. Lehman. The next question that comes in is, are there other areas within bleeding disorders where you could foresee mathematical modeling to be helpful?
Yes. I mean, I guess other areas. I mean, I think one thing we're trying to use the model for now also is for thinking about like different pharmaceutical treatments and things, and trying to understand how they work in the system, different antibodies to different things. So we're looking into that. And really, we don't know yet what's going to happen when we look at hemophilia B and C. And I know there's some really interesting questions in hemophilia C because it's just not entirely clear that all people with hemophilia C bleed a lot. And so we don't understand that. And I think there's a lot of room to work in this space, other than just looking at, say, hemophilia A. But yeah, I think there's a lot of things we can look at with the model, BWD and the hemophilias and et cetera.
Great. Great. We've got one last question that's come in, and I think this is a good one
Okay.
to end it on. It's what was the drive or reason behind this project and research?
That's a great question. So I started working with a collaborator many years ago. Keith Neves, his name's here on the slide. He's a chemical engineer slash bioengineer. He is now in a bioengineering department at the University of Colorado, and he worked very closely with Jorge de Paula, who is an excellent researcher in the area of thrombosis and hemostasis, and had worked in the hemophilia space for quite some time. And the two of them together were looking for, you know, other interdisciplinary approaches to try to answer some of these questions about phenotypic variability that they'd observed just in some of their own research and with their collaborators and at the hospitals where they worked. And so they kind of came to me and Aaron Fogus and Suzanne Cindy, the other two mathematicians on this project, and sort of posed this question, like, is this something that your models can work on? And we'd been working on models of clotting to really more focused on kind of understanding the biophysical and biomechanical mechanisms within clotting, just because there hadn't been a lot of work done on understanding how flow in cells really affect the process. And that had been our main focus. So this kind of allowed us to step out of that and be a little more focused on looking at bleeding disorders. And that led us down this road of looking at the statistical analysis and so forth. So I think it was more, meeting people and forming collaborations with people that worked in the space of bleeding disorders. And it's just been so wonderful to work with all these people and learn from them over the years. But that's really how it got started.
Great. So a great last question, I think there. Thank you for explaining that. So we really I'd like to, just take the time to say thanks, Dr. Wiedemann, for joining us this afternoon. We appreciate, your time and expertise in this area. And this was a very well presented presentation. Thank you very much.
Thanks for having me. Thanks so much. It was fun. I wish I could see more people, but thanks everybody for listening.
I think we all need that at this point, to see more people here. But
I know.
Please note that.
Well, it was fun to be here.
Yeah, no, thank you. Please note, everyone, that the recorded webinar will be available on Friday, June 4th. You can find it at hemophilia.org under the events tab with all of our other archived webinars. Also available in the events tab is our upcoming schedule for our weekly Wednesday webinars. Thanks once again, Dr. Wiedemann, and everyone have a great afternoon.
Thank you. Have a great day.